Hi. In the last module what we have seen? We have seen how we can design a drug screening device using microengineering, right. So, if you recall, we have seen how to draw a process flow using photolithography for fabricating a microheater. Over the microheater there was an insulator, over insulator there were interdigitated electrodes, and over the interdigital electrodes we created using a separate process a mold to form channels in PDMS, and we have done the bonding of this PDMS with channels over the interdigital electrodes below which there is an insulator, below which there was a microheater. So, how the chip will look like and how we can use this chip to understand the or to evaluate the efficacy of different drugs, let us see today, right, and we will take some examples that what kind of cells cancer cells we can load into this chip, and what when we flow the drug what kind of diffusion occurs, and because of diffusion if the drug is effective whether cancer cells dies or not, and if it dies what kind of electrical response we obtain. Right. That is the idea, to create an engineering device that can be used to understand the efficacy, the performance of the given drug, and the application here is cancer. Okay. So, if you see the screen, what you see here is last time we have seen how we can fabricate, or the process flow for fabricating a heater, process flow for fabricating interdigitated electrodes, process flow for forming channels in PDMS. Then we have stick the PDMS to the interdigital electrodes and the final microfluidic chip will look like the one shown in figure here. This is the actual photograph of the fabricated microfluidic device. So, like last time we discussed, right, that in if you see this one, right, take one single channel, what we have? We have a longer channel and we have a shorter channel. Here we have a pillar. Now, we will load cancer cells with matrigel, right. If you remember what we have discussed, that a cancer, a tumor is surrounded by extracellular matrix ECM that provides nutrition and required things for tumor to sustain and in fact grow. So, how can we replicate this in vivo situation? This is in vivo within the body, alright, onto the in vitro platform. This platform is called in vitro or in vitro. This is what happens within the body, it is called in vivo or in vivo. Okay. How you pronounce does not matter, it is written as in vivo, in vitro. Okay. So, inside the laboratory if we are growing a cell, so if we are growing the tumor, if we are performing experiments which trying to mimic the actual situation which happens within the body, then this is called in vitro system, and microfluidic chip mimicking the in vivo system, here we are looking at it. So, what we have to do? We have to add cancer cells within the with matrigel into this shorter channels. Okay. So, now we have lot of cancer cell with matrigel. Then what we will do? We will flow the drug. So, we have peristaltic pump. So, there is a reservoir from which the drug will come in, and the drug that goes out will again go back to the reservoir, right. This is where it stores the drug. Within the peristatic pump we can store the drug with media. Of course, we cannot just flow the drug, right. What do you mean by flowing the drug? When we take the drug, it will flow through the it will mix with the blood and flows with the blood, right. So, the role of the blood here, we are replacing media as a blood and we are adding drug to the media, right. So, when I just flow media in this channel, in this particular channel like this, right, and the media will diffuse into the matrix gel with cancer cells, and I may get let us say impedance Z1. If I am measuring the impedance, why I can measure impedance? Because there are electrodes in this area, right, like this there are electrodes in this area. So, I can measure impedance Z, right. So, what happens when I flow the media, then I will have impedance Z. If I flow media with drug, then the cancer cells may die. If it dies, then the conductivity would increase. If the conductivity increases, impedance decreases. So, I will have a different value of impedance Z2. If the value of Z1 and Z2 are significantly different, there is a statistical significance, then I say that my drug is effective. So, let us see. So, this is what our concept is, right. Our concept is that the current regimen, the current treatment regimen is you give the chemotherapy, right, for a given tumor, you give the chemotherapy, wait for certain time, right, and if there will be reduction in tumor size if the chemotherapy is effective, right, but the cells develops adaptive resistance and there is a regrowth of tumor. Now, this whole process to know whether chemotherapy is working or not takes ideally few months. So, the problem here that we are addressing is that how can we design a device that can tell which drug would be effective from a given number of drugs, for given number of let us say A, B and C drug for a particular patient P1, which drug would be effective, A, B or C, right. We do not know and current techniques takes about few months. So, how about we create a microfluidic chip, right. From patient 1 we take the cells, right, we take the cells from tumor region, right, we load into the microfluidic chip and we test A, B and C separately, and we can then find whether which drug is effective based on the electrical properties of the cell. So, if you see here there are gel inlets, drug inlets, impedance measurement, because the idea here is that now how can you, when you load the cells would die, right, because it is not at 37 degrees centigrade. That is why the bottom of the chip is nothing but our heater, right. The bottom of our chip is our heater. Over the heater there is insulator, right, and then we have electrodes, correct, we have electrodes. Eight different electrodes, right. So, one electrode, second electrode, third electrode and 8 electrodes, right. So, we can have here 8 electrodes, you can see here impedance measurement, right. So, with the help of electrodes we can measure the impedance. Now, when you load the, when you bond the PDMS with channels onto this microfluidic chip. So, you assume like this, there is a microfluidic chip with heater, there is an insulator, there are electrodes, right, and you are bonding this chip with PDMS, interdigital electrodes, micro heater, and this is your substrate. Substrate is glass, right. So, when you do this, what will happen? So, these are, you can also form interdigitated electrodes using this particular pattern, alright. So, you can see here 3D schematic, your A would be a 3D schematic of the platform for multiplexing drugs susceptibility testing, B would be the, this one would be cancer cells are placed in a 3D matrix, right, in a 3D matrix. C would be cancer cells are intact and viable in 3D gel structure. You can see here cancer cells are intact and viable in a 3D gel structure before introducing drugs. D would be the intact cells are reduced after introducing, you see here the number of cells that we are presenting are reduced after introducing the effective drug flow, right. This is actually fabricated device and this is the interdigitated electrodes, E is interdigitated electrodes onto the device, right. So, the point that we are making here is that this is a conceptual diagram, a 3D schematic diagram of what, of telling our hypothesis that if we have this structure and if we load the cells with some gel and we introduce a drug, right. So, B is our microfluidic chip, C is where we are introducing these cells, D is where we are introducing drug and the cells are dying, right, and if the cells dies then we can see change in impedance. This is our schematic representation. So, now let us see actual experiment. Now, always remember, whenever you create or whenever you fabricate or whenever you want to fabricate a microfluidic chip, always you need to perform simulation. Why simulation? You need to understand how is your flow, right. So, to understand microfluidic chip in detail, you need to understand lot of things like Reynolds number. One of the example is Reynolds number, right. What is the shear stress within the microfluidic channel, right. What is the velocity, right, whether the flow would be smooth or not. Okay. So, a lot of things you need to understand and that is why we perform a simulation. Now, after performing simulation, you see here the plot of normalized impedance versus frequency. Now, what I said is we will measure the impedance, that is why you require a impedance analyzer. Okay. And we will be looking at all the things that we are talking in the series of lectures like impedance analyzer, peristatic pump, microscope, right, different kind of microscope, whether it is inverted microscope or it is a metallurgical microscope or it is a stereo microscope, we will look at it. We will look at the micro manipulator, right, we will look at the incubator, we will see PDMS bonding onto the electrodes, onto the glass, right. So, this process we will actually see in this particular course. Right now, let us understand how this device, how this chip can be used for testing the drugs. So, if you see here, we have several controls, 3 controls in particular. One is just gel. When you introduce into this smaller channel, when you introduce, so if you see this particular thing, right, then when you introduce just gel in this channel, okay, what is the impedance? Then when you just introduce gel plus drug, so there is no cell, okay, there is no cell, thus gel is there and you are introducing here drug. So, gel plus drug, what is the impedance? Now, you have gel plus cancer cell, no drug. So, here the third case will be gel plus cancer cell, right, control 3. What is the impedance? These are our controls. Okay. Now, fourth control, we have used 4 control, I am sorry, we have used 4 control not 3. So, fourth control would be gel plus cancer cell and media flow. So, now we are flowing just media, no drug, no drug. Okay. Fifth would be, now we have cancer cell which are breast cancer cell, and we are loading media plus drug. What is the impedance? We are flowing media plus drug, and here it will be gel plus cancer cell, which is breast cancer cell. Next test would be if it is a tolerant breast cancer cell, right. One is susceptible, which will die with the introducing different drugs. Another breast cancer cells are tolerant to it, resistant to it, but resistance we cannot say, we can here say that it is tolerant to the particular drug. So, when we treat the breast cancer cells with a drug and leave it for some time, it becomes tolerant. So, instead of susceptible, we will use breast cancer cells which are tolerant, right. So, gel plus breast cancer cell which is tolerant breast cancer cell, and we will flow media plus drug, and we will see the change in impedance. So, when we perform this experiment, what we found is that at 1000 hertz, at 1000 hertz, we could see maximum difference between control and cancer cells, between control and actual experiment, that is our drug susceptible breast cancer cells when we flow the drug, and breast cancer cells tolerant one when again we flow the drug. So, if you magnify this, right, if you magnify this, we can clearly see here that there is a difference between all here 4 controls, right, these are 4 controls. So, cancer cell plus media flow you can see here, then you have gel control 4 here, right, control 1, control 2, control 3, and then we again have gel 1, control 3, control 4, right. So, what we see here is that the maximum impedance magnitude shift occurs at 1000 hertz. We just were interested in understanding at what frequency we can see the maximum change in the impedance magnitude. So, what we found is 1000 hertz is what we have to use. Now, we also need to understand why we cannot use a static platform. So, what do I mean by static platform? Static platform means if I have a well, if I have this micro heater, right, if I have insulating layer and over there are interdigited electrodes, can I just put matrigel with cancer cells, right, and I will get Z1, and if I put drug 1 let us say with media, right, drug 1 plus media, then after some time let us say 24 hours I would be able to see a difference Z2 if the drug is effective, correct. So, this is a static study, static way of measuring the change in impedance. Why I cannot perform this experiment, or why should I not perform this experiment, right, that is a question. So, the answer could be that because our body is dynamic and not static, we should try to see what happens in a dynamic situation compared to a static platform, right, because here the drug plus media is in continuous contact with your cancer cells that are loaded inside the matrix gel. So, static platforms will give us a different response compared to dynamic platform, and we should be looking at dynamic platforms because our body is dynamic, right. So, that is why we are understanding this microfluidic chip for rapid drug screening. Why I said rapid? Because you can have 8 different channels, you can have n number of channels in reality. So, you can test at a time several drugs for a particular patient, right, and within 24 hours range you should be able to tell which drug is effective for that particular patient. So, we are designing a patient centric platform, we are designing a rapid drug screening platform which is patient centric, right. So, if you see the screen, what you see here in the screen is that we are looking at electrical response of drug which is susceptible as well as tolerant, right, for murine cancer cell. So, if you can see the screen, we are talking about murine, right, cancer cell that are taken from the mice. Okay. Now, what we see here? We have control 1 which is gel, control 2 gel plus drug, control 3 gel plus cancer cell, control 4 gel plus cancer cell media flow. This is experiment 1, this one is our experiment number 2, right. So, what is our experiment number 1? Gel plus cancer cell plus drug in a static situation. Experiment number 2 is gel plus cancer cell plus drug in a dynamic situation, all right. So, first let us see drug susceptible breast cancer cells. Here we have tested 3 different cell lines. First one is breast cancer cell line, second one is melanoma cell lines, melanoma is skin cancer, third one is prostate cancer cell lines. Okay. So, 3 things we have tested: breast cancer, melanoma and prostate cancer. Now, let us see. So, if you see at 0 R, right, there is the impedance is almost 1 which is normalized impedance, right, and as you keep on increasing the R's from 0 to 3 to 5 to 7 to 9 to 12, you can see a change in impedance for our experiment which is static, which is here, and dynamic. So, this top one is dynamic, the bottom one is static, right. We can clearly see that a dynamic experiment shows us different result compared to static experiments, right. Now, what if we have drug tolerant cells? What if we have drug tolerant cells? In that case also we could clearly see a signature at 1000 hertz, this is impedance, right, and we can see that the static results are different than dynamic. However, we can clearly understand effect of drug for using dynamic platform. What if we are using melanoma? In melanoma also we can see a signature. What if melanoma is tolerant? Drug tolerant melanoma cells also we can see a particular signature. You can very easily see, right. So, you have different signatures for breast cancer cell which is susceptible, breast cancer cells which are tolerant, right, melanoma cells which are susceptible, melanoma cells which are tolerant. Now, you will see we have used a particular drug called carboplatin, correct. So, for breast cancer cells and melanoma cells generally platinum based drugs are used for chemotherapy, platinum based drugs are used, while for prostate cancer Taxol based drugs are used, Taxol based drugs. That is why when we go for prostate cancer cells to understand the effect of drug, we are using paclitaxel, right. So, anyway, this is just for understanding that platinum drugs generally are used for breast cancer cell and melanoma, while Taxol based drugs are used for prostate cancer, right. And our interest is not to really go into details why Taxol based drug, why platinum based drug. Our understanding for this subject lies into the fact that can we design or can we fabricate a device that can perform rapid drug screening for a given number of drugs and can tell for a particular patient which drug to administer or which drug to give. So, again for prostate cancer cells you have a different signature. Now, there is one more thing that is interesting. So, the change in the impedance for tolerant cells is different than change in impedance for susceptible cells, right. From here it may not be easily visible, right. From here it may not be easily visible, but we can see in the next slide that you can clearly see a difference between impedance, normalized impedance when you are considering susceptible cells and when you are considering resistant cells. So, for the present study these plots are obtained using 41 mouse breast cancer cells, B16 F10 mouse melanoma cells and DU145 drug resistance human cells or prostate cancer cells, DU145 prostate cancer cells. Okay. Now, if you see here what we obtained? We obtained a clear signature, when we are plotting normalized impedance versus time for impedance measure at 1000 hertz, because that is our optimized frequency. When we plot normalized impedance versus time for drug susceptible and drug tolerant breast cancer cells measured at 1000 hertz frequency, we can see that the drug tolerant cells, the impedance change is less compared to drug susceptible cells. Red one is drug susceptible, blue one are drug tolerant cells, all right. Same thing goes for melanoma, drug tolerant which is this one, blue one, and drug susceptible which is red one. Same thing also goes for prostate cancer. But one thing that we need to understand is that prostate cancer cells, particularly resistant, are very difficult to kill. The resistant, drug resistant prostate cancer cells are extremely difficult to kill. In general, prostate cancer cells are difficult to kill with paclitaxel, and we could see. So, these are all electrical measurements, right? A, B and C, A, B and C, all three are electrical properties of a cell that we are interested in measuring for different drugs, correct. And what we can see here that A, B, C normalized impedance magnitude for drug susceptible and tolerant or resistant breast cancer, melanoma and prostate cancer cells at drug concentration of 30 micromolar at 0 hours, 3 hours, 5 hours, 7 hours, 9 hours and 12 hours time interval, right. So, this is for 30 micromolar concentration. Now, what are these D, E and F? We have to understand whatever signatures we are obtaining, whatever methods we are using are correct or not. So, how can we test it? So, we can test by understanding whether the cells are dying or not. So, if the cells are dying, then the cell count will reduce, right. If the cells are dying, the cell count will reduce. So, what we can do? We can extract the matrix gel with cancer cells after the drug is flown. So, let us say this is cells with matrix gel, right, and these are cells within matrix gel after the drug is flown. Right. How many cells have died? How many cells have died, right, in terms of percentage. Now, how we will know how many cells have died? So, we know when we initially load into this well, how many cells we are loading. So, is it 25,000 cells per milliliter, 25,000 cells per milliliter, or it is 40,000, we know, right, whatever number we are loading we know. So, this would be our 100%. Now, when we recover these cells after the drug is flown, after drug is flown, if we recover these cells and count it using hemocytometer, hemocytometer is used to count cells live or dead. So, using hemocytometer we can count number of cells. So, when we perform this experiment of counting number of cells after the drug is flown, then we found that you see here clearly drug susceptible cells, and this is drug tolerant and this is drug susceptible. Drug tolerant and drug susceptible cells it reduces as time increases for when the drug is flown in the channel, right. Same thing goes for melanoma and prostate cancer. And we can see that this signature number of cells dying, right, corresponds to the impedance, is not it? That means whatever impedance we are measuring has a correlation with the number of cells dying, because when a cell dies the conductivity would increase. When we lyse the cell, it is called lysis, dying or lysing. When you lyse a cell or when lysis happens, the cell with nucleus will rupture, So, the content within the cells will come out. What are this content? Can be potassium, can be calcium, can be other proteins, right, can be different things. Now, because the contents of the cell will come out, the conductivity would increase and impedance would decrease, all right. Now this particular technique is not so sensitive that it can measure the one cell dying, it cannot measure single cell dying, okay. Group of cells should die, and then we have to understand how many cells are dying to understand the sensitivity of the platform. However, for the given study we could clearly see that there is a correlation between number of cells dying and impedance changing. So, here what D, E and F shows? D, E and F shows cell viability of a drug susceptible and tolerant resistant breast cancer, melanoma and prostate cancer cells after flowing drug for 0 hours to 12 hours, right, in a interval of 2 hours. First is 3, 5, 7, 9 and 12, 2 to 3 hours. So, when you load the cells in hemocytometer, you have to mix the cell with trypan blue dye, right, to understand which cells are dead and which cells are alive. This is a very basic technique in biology to understand or count the number of cells. Now, after doing this we can also test our our results, or we can compare our results with a gold standard, which is our MTSSA, cell viability. Cell viability, you see here, this is cell counting, this is cell viability. Okay. So, here cell viability, how can we understand? Using a technique called MTS study, MTSSA. So, when we perform cell viability, we can again see that there is a signature when the drug tolerant cells and drug susceptible cells are measured, right, for different concentration of drugs. The are measured for different concentration of drugs, and there is, as you increase the drug, the cell starts dying, cell starts dying. So, when we perform all these experiments, we can say that the impedance measurement can be used to understand whether the drug is effective or not, correct. From this, all these experiments, we can say that impedance measurement can be used to understand whether the particular drug is effective or not. So, let us quickly recall what we have studied in this particular class, right. We have concentrated on developing a microchip for rapid drug screening. So, the idea is, can we develop a drug screening tool which is patient centric, and we can see the change, or we can understand the efficacy of drug within 12 to 24 hours. For that, we have seen how we can draw a process flow for fabricating a microchip. We have seen process for individual components, starting from heater, then we studied interdigitated electrodes, then we studied how to create a PDMS channel using a mold, and here we have considered SU8. Then when we have all three together, right, how it will look like, right, how it will look like when we have all three things ready, and when we bond it, when we bond the PDMS with glass, right, how it will look like. We have seen this, right, which is our in vitro platform to understand or to measure or to evaluate the efficacy of given drugs, right, this platform. After that, we understood how we can plot a 3D schematic to convey our message, what and why this technique we are using, and what is our hypothesis. Then we have seen actual results, and we found that 1000 hertz is the optimized frequency for our experiments. Then we have seen that for given controls and given experiments, right, how the impedance, normalized impedance changes with respect to time, right. And here we can clearly see that the results obtained from static platform is different than results obtained from dynamic platform, right. Also we have seen that we can measure not only the breast cancer susceptible cells, but also breast cancer tolerant cells, and same thing for melanoma susceptible and melanoma tolerant cells. We also tested prostate cancer cells, which are human prostate cancer cells, and number is DU145, and prostate cancer cells that are resistant to drug, which is and we have seen the plot. Similarly, for breast cancer, what we have seen, breast cancer cells we have used 4T1 mouse breast cancer, and for melanoma B16F10. So, here if you see the signature, we can clearly see that as if the drug is effective, right, for a given drug, we can clearly understand that the difference in the impedance, right, for for when we pass the drug for about 12 hours. And we can also see that the drug resistant cells, particularly prostate cancer, we do not see that much change in impedance, because it is very difficult to kill prostate cancer cells using paclitaxel, right. We have also performed and we have seen how the plot will look like for cell counts using hemocytometer. Finally, we have understood cell viability study using MTS assay, right. So, if now if we are given so if we are given what exactly you will be what kind of experiments you will perform, or what kind of device you will design, right, then you can now tell that yes, for cancer, for chemotherapy, we know a microfluidic platform can be a potential answer, right. A microfluidic platform can be a potential answer, because we can measure different drugs at the same time by measuring the change in impedance. That is, we can now design a patient centric platform using our knowledge of microengineering. How to measure the efficacy or performance of drug? By loading the cells into the well below which there are electrodes and passing the drug, and these cells are within matrigel, and when the drug diffuses through this matrigel, it will kill the cancer and you can see the change in impedance. Now, you can do for one channel, you can do for 8 channels, which we have seen in this example. That means 8 different drugs for one particular patient you can use, and you can say which drug to give to that particular patient, or 8 different patients, 8 different cancer cells, right, or 8 different patients suffering from a particular cancer, you can load, the cells from the patient you can load into this microfluidic platform and pass a particular drug and see whether the drug is effective for which patient. Again you can save the time, right. The idea of understanding or proposing or performing research in this area is, can we reduce the time, which is currently used to administer a particular drug regarding chemotherapy for a given patient, right. Can we reduce this window? Can we bring it to 24 hours? Can we bring it to 48 hours? And can we propose a particular drug, right. So, we will be learning this kind of interesting microfluidic chip. I hope it is interesting to you. I hope it is new thing for you to learn, that this micro technology is not only meant to understand micro sensors that can be utilized in your display, in your iPad, in your Android or in your laptop, right, or even in satellites. Micro technology can also be used for clinics. Microtechnology can also be used for creating a novel biomedical platforms. That is the beauty of microengineering, right, that it is not limited to a particular area or field of focus. It can be utilized for several applications. If I want to make a gas sensor, I can make it. If you want to make an accelerometer, you can make it. If you want to make a pressure sensor, you can make it, right, or you want to make a microfluidic chip, you can make it, because you know the basic, the understanding, right, of microengineering. So, we will take another topic, interesting topic, and what we will be covering in these lectures, right. This will be, I think it is not available somewhere, most of the topics are not available in some particular book. So, the examples are the current research area which lot of people are interested in and lot of groups are working in. Alright. Along with that, we will be keeping our focus on to the understanding microengineering, and we will be taking you to the laboratory and show you few of the equipment that I discussed in this particular module. Till then, I will see you in the next class. Take care, read the lecture. If there is any question, you can ask us in the forum. Bye.